So welcome to another Chapter 6 virtual lecture on microbial growth. I focused the last lecture on the chemical requirements for growth. Now I want to talk about the physical requirements for growth. We're going to talk about how temperature impacts bacterial growth, and pH, and osmolarity, which we know is related to water transport. Let's first talk about temperature. Now, in order to understand how temperature affects the growth of any cell, we have to understand that parts of the cell are temperature sensitive. For example, proteins are temperature sensitive. If the cell is at temperatures above optimal, the proteins begin to lose their activity. Now, the reason proteins lose their activity is due to a process called denaturation. They denature, they unfold, and all that means really is their structure is unwound, and because the structure is no longer held in place, their activity is lost. So you can imagine then a cell without proteins is a cell that's dead. We also know that lipids are impacted by temperature, which are a very important component of membranes. So if the temperature becomes too low, membranes become rigid and they can actually crack like ice would crack, causing the inner contents to spill out. Conversely, if the temperature gets too high, membranes become too fluid, and what happens is the inside of the cell and the outside of the cell mix together. So we have to understand that these molecules that construct a cell are sensitive to temperature, and then as an extension of that, cells are temperature sensitive. So here's the effect of temperature on growth. Now here, we've seen figures like this before. Here's the rate of growth on the y-axis. Here's the temperature increasing on the x-axis. Now the first thing you notice is I've given you no values here because this depends on the bacteria. But here's generally the effect of temperature on a cell. We have, let me point out this temperature here. That's optimal temperature. Notice at this given temperature, the growth rate is at its maximum. Now, as we begin to deviate towards either warmer or colder temperatures, notice how the growth rate decreases in both cases. Let's first talk about the effect of higher than optimal temperatures. Notice the sharp decrease in the growth rate. We see that the growth almost falls off a cliff. It doesn't take much deviation higher than optimal till we reach what we call the maximum temperature that supports growth. This maximum temperature that supports growth is essentially that. It's the temperature at which we reach the bottom of the growth rate. Anything over maximum, the cells are dead. Notice again, it doesn't take much deviation to reach maximum. Notice as we deviate colder than the optimal temperature, notice it takes a large deviation in temperature to reach the minimum growth temperature. So cells can withstand changes in temperature on the cold side of optimal. However, on the warmer than optimal side, we see a sharp decrease. So again, what's happening here? As we go above optimal, proteins lose their activity. And at above this temperature, all the proteins no longer work, therefore the cell is dead. As you go lower than optimal, recall that lipids are most affected. Lipids become rigid. Membranes become fragile. Let's think big picture here, folks. This is why we cook food. This is why we put things in the refrigerator. Right? When you cook food, what you're doing is you're raising the temperature above optimum, so any bacteria that may be contaminating that food product are all killed before you consume it. Makes sense. Conversely, by refrigerating food, we're slowing the growth of bacteria, and by doing that, we're not killing the bacteria, but essentially what we're doing is we're preventing spoilage. We're preserving food when you go at temperatures below optimum. So even though I've introduced a new concept here, I think you've been practicing and examining this idea in your daily lives in relation to food. Here's an example of bacteria growing on a plate at 3 different temperatures. If I were to ask you, and a question's going to pop up on your screen, what temperature appears to be optimal for this bacteria to grow? Yeah, if you look at this, I would say 37 degrees. That's where we have the most growth. That's where we have the most number of colonies. Notice then on this graph, 37 degrees is right here. And as we deviate, as we go down in temperature, notice that the bacteria still grow at 30 degrees, but they don't grow as well. And if you even go further down, notice what happens to the growth rate. The bacteria still grow, but they're growing at a much slower pace. Therefore, there are less bacteria on that plate. So what we're showing here are temperatures from optimal to less than optimal. Now, we can often classify bacteria based on their optimal growth temperatures. I want you to learn these 4 groups. I'm going to start here with mesophiles. And the reason I'm starting here is because most bacteria fall within this group. So let's define what a mesophile is. A mesophile represents a bacterium that grows best at 37 degrees Celsius. That's optimal. Now that's interesting. I don't know how many of you have worked in centigrade, but do you know the body temperature of a human in centigrade? Yeah, the body temperature of a human in centigrade is 37 degrees Celsius. Mesophiles grow best at 37. Humans are at 37. Those bacteria that are pathogens, and even those that are non-pathogenic that live in our bodies, all of them fall in that mesophilic category. You see, there's a reason that humans have more bacteria on them than they have their own cells. And we are a perfect environment for bacteria in this group to grow. Therefore, mesophiles like to live in humans. Notice here their minimal growth temperature is around 15 degrees Celsius. Their maximum growth temperature is around 45 degrees Celsius. So can you guess then why a fever provides some benefit to a human? That's right, the body responds with a fever because as our temperature raises, we're going to slow down the growth rate and allow our immune systems to catch up. And so now our immune system can start to deal with that infection. Can you also see why refrigerating our food is important? As we refrigerate our food, those bacteria that are mesophiles can't grow, so we're suppressing their levels and preventing spoilage. Now, does refrigeration work on this group called psychrophiles? The answer, of course, is no. Their optimal growth temperature is somewhere between 10 and 12 degrees Celsius. A refrigerator keeps your food at around 4 degrees Celsius, which we can see is permissible for psychrophile growth. So that should concern you, right? If psychrophiles can grow on the food that you refrigerate, when we consume that food, we're going to get sick. Would that be an accurate statement? Of course, the answer is no, that's not accurate, because notice the maximal growth temperature is 20 degrees. So when you consume that food, your body temperature is not permissible for that bacteria to grow, so you don't worry about that psychrophile getting in your system and causing damage. So now let's go on the other side of this. We have thermophiles and hyperthermophiles. Thermophiles can grow at higher temperatures. Notice here we have a maximum of about 68 degrees Celsius for a thermophile. And for a hyperthermophile, see, we have an optimal temperature at around 93 degrees Celsius. So again, should we worry about thermophiles and hyperthermophiles making us sick? The answer, especially for hyperthermophiles, is no, because notice the minimal growth temperature far exceeds body temperature. And even if you examine the minimum growth temperature for a thermophile, you can see again it's below that which is of human body temperature. So we tend to find thermophiles and hyperthermophiles in places like volcanoes, hot springs, in those very unique environments in which most living things cannot grow. Humans cannot withstand temperatures in this range, but yet bacteria can. So this is why we can find bacteria in almost all places on the planet, because they can grow at such a wide variety of temps. So now let's test your understanding on this. We have a mesophile. We have an optimum temperature of 35 degrees, and that is the temperature in which we're going to grow the bacteria. So now, which line is going to best represent a mesophile growing at its optimum temperature? Press pause and log in your answer. That's right. A is going to best describe the growth of a mesophile at its optimum temperature, because notice the rate is the highest. The steepness of the slope represents the greatest change in the number of cells over time. Let's just try this out. I know it's not in the poll. What if I were to grow a mesophile at 60 degrees Celsius? Which line would represent the growth of a mesophile at 60 degrees Celsius? Answer in the screen that pops up. That's right, the answer is C. Because if you go back and look, a mesophile at 60 degrees cannot grow, therefore the number of cells will not change over time. So we're going to have many questions like this on the exam where I give you growth curves and I'm going to ask you a question and you're going to have to apply that physical condition or that chemical condition to the growth of that bacteria. Now let's move on to pH. Now most bacteria are going to fall in this group called neutrophiles. That group of bacteria grows best between 6.5 and 7.5, which happens to be a neutral pH. Does anyone know the pH of your blood? That's right, the pH of your blood is 7.2. Again, can you see why we are the perfect vessel for bacteria to grow? Not only are we the ideal temperature for most bacteria, we also have the ideal pH for most bacteria. However, there are some bacteria that are considered acidophiles, which can grow in acidic habitats. So why would we be concerned about these bacteria and even another group called fungi? Well, if we try to preserve food, for example, we often add acids to food to preserve them. Well, these bacteria can grow there. So these acidic waste products that we use to help preserve food are going to work very well for neutrophiles. Therefore, we use acids to prevent food spoilage. The group that I would be concerned about are these acidophiles, of which they're not a big group, but it's a group nonetheless, in which can grow in these acidic habitats. So let's think about this. What growth curve would best be depicted by a neutrophile growing in our stomach? A, B, or C? Press pause and answer the question. Yeah, that's right. The correct answer would be C. Now, you needed to have some background knowledge. Whether you took A and P or not, we should understand that the stomach has a pH of about 2, which is extremely acidic. Therefore, the stomach acts as a protective barrier because neutrophiles cannot grow in your stomach due to the extreme pH. So now we see pH can affect bacterial growth. Water is also going to be important. Now we know water is a major component of the cytoplasm of any cell, and therefore all of the cell functions for a prokaryote or most of the important cell functions are going to occur in that cytoplasm. So as water changes in a cell, it's going to affect the internal environment. Most cells are going to die when they dry out. So in the absence of water, what's going to happen is water is going to rush out of the cell, and it's that drying that causes those cells to die. Now, some cells have evolved to get around this. For example, the genus Mycobacterium, which is a genus of bacteria which is responsible for diseases like TB and leprosy. We talked about this in Chapter 3 and in Chapter 4. Their cell walls are high in this waxy substance called mycolic acid. Therefore, these bacteria can retain water, so they don't dry out as easily. This is why somebody with TB, if they cough, that bacteria can stay airborne for longer periods of time because the dry air will not be able to easily suck water out of that bacteria. Some bacteria also produce endospores, which allow them to survive a dry environment. Genera of bacteria that produce endospores, you may remember, are Bacillus and Clostridium. Now, what are two of the physical effects of water? How can water have an effect on cell structure? We want to talk about osmotic pressure and hydrostatic pressure. Now, let's finish up the lecture talking about osmotic pressure. If you recall, osmotic pressure is due to the amount of solutes across a membrane. So, for example, if I put a cell into a hypertonic solution, so which way does water move if the cell is bathed in a hypertonic solution? That's right, water moves out. What's going to happen to the size of the cell if water is moving out? That's right, it's going to shrink. It's going to lose volume. And as we just discussed, a cell needs a certain amount of water to carry out its cellular functions. So if a cell dries out in a hypertonic solution, the cell is going to die. Now in a hypotonic solution, the environment has less solutes than the cell itself, so that's going to pull water into the cell. Now what's going to happen is those cells begin to swell. Now is this true? Cells placed in these solutions will swell and burst. Is that true for bacteria? And if it's not true, explain why. That's right, bacterial cells don't explode because they have that rigid cell wall. And so that's an advantage for bacterial cells compared to a human cell which lacks that cell wall. So as we just stated then, salts and even sugar in the environment directly affect osmosis. And this is going to be important as we go forward because there's a group of bacteria called halophiles. Now let's define what a halophile is. A halophile is a bacteria with the ability to grow in high salt. So here's an outline of what happens to a cell in different solutions. In an isotonic solution, for every water that moves in, a water moves out. So there is no change in the size of the cell. Now, if you were to put a cell into a hypertonic solution, because there's so many more solutes outside, water will be sucked out, causing the cell to shrink. And when the cell loses too much volume, no longer can it carry out its essential functions, and therefore a cell can die. Now we know because of a cell wall, a cell placed in a hypotonic solution, which there are less solutes outside than inside, that's going to suck water into the cell. But the size of the cell is directly impacted then by the cell wall itself. So we won't have overexpansion. So all bacteria have a level of protection due to their cell wall. But how about this case with a bacteria in a hypertonic solution? Well, some bacteria can actually regulate the loss of water. They're called halophiles. What they essentially do is they can control the salt content of their cell cytoplasm. Therefore, they don't lose as much water. Now, a great example of this, and one you should know, I want you to learn this, commit it to memory here. Staphylococcus aureus is a specific bacteria that is a halophile. It can grow in high salt, but it's facultative in nature. That means it can grow in low salt, it can grow in medium amounts of salt, it can grow in high salt, it can adjust to its environment. Now, when I show you this later on in the semester, I hope it makes sense. Staphylococcus aureus is a common inhabitant of our skin. And what do you know about the salt content of your skin? That's right, the salt content of your skin is rather high. So how can these bacteria continue to survive and grow? Well, they can do that because they're halophiles. So that's a very important class of bacteria. Now that's going to include our discussion on the physical requirements. We've walked through the temperature impact on growth. We've walked through the pH impact on growth. We walked through the impact of salt and sugar on growth. If I were to give you one last example here of why halophiles are important, we often use salt and sugar to preserve food. Ham is salted. And the reason being is it won't spoil because most bacteria can't grow in the high salt. However, salt preservation will not necessarily stop food poisoning caused by Staph aureus due to the fact that halophiles can still grow. And that's a concept we're going to see in the lab. So that concludes our discussion on the physical requirements on microbial growth.